The overall goal of this procedure is to study the chemosensory physiology of individual PEG sensilla on a scorpion pecten. This is accomplished by first immobilizing an anesthetized scorpion, ventral side up, on a glass slide using modeling clay. To set up requires submerging the pectins in a custom-made oil-filled chamber and making electrodes filled with a chemical stimulant. Chemosensory responses are recorded at individual sensilla on the pecten, which ultimately produce similar response profiles to aqueous chemostimulants.
We used to record from the basis of sensilla while blowing volatile stimulants across the pores. The main advantage of the tip recording technique is that it allows the stimulation to be isolated to one PEG sensillum without affecting the activity of its neighbors.
This method can help us answer key questions in the field of chemosensory biology. For example, why do animals use so many repeated sensilla in chemical detection and perception?
Scorpions might not seem like the safest animals to study. However, not all species are dangerous or difficult to deal with. For example, the desert grassland scorpions are only 4 to 5 centimeters long, are easy to maintain in the lab, and rarely sting if handled properly. If stung, the venom artificially triggers your sensory neurons, some of which transmit pain information. The sensation usually subsides in about 15 minutes. Scorpions are hardy subjects and can endure multiple days of experimentation, often being returned to their habitations when the experiment is complete. To immobilize the scorpion, place it in a pre-chilled glass container. Then place the jar containing the scorpion into a freezing environment for a minimum of 2 minutes and remove it from the freezer when the animal is motionless. Now place the immobilized scorpion ventral side up on a microscope slide. Secure the stinger, tail, legs, and pedipalps with moldable clay. The scorpion's pectins are paired, comb-like structures near the sex organs and are unique to these animals. A chamber must be made to contain a single pecten without removing it from the animal. First, make a platform for the pecten to fit onto. A glass cover slip, cut to approximately 5 mm by 18 mm, usually serves well. Second, adhere double-sided adhesive tape to the platform and trim the tape to fit if necessary. Third, melt some 100% beeswax chips in a glass dish using a hot plate. Now dip 3 of the platform's 4 edges into melted beeswax to a depth of 3 to 4 mm. The cooled wax serves as a wall to make a chamber. Now place the unwaxed edge of the platform just posterior to the point of pectinal insertion on the animal. Stabilize the platform by gently pressing its right and left peripheral edges against the clay used to restrain the animal's legs. Using fine-tipped forceps, carefully hold one of the pectins along the pectinal spine and gently bring it out from underneath the cover glass and lower it onto the adhesive platform floor. Stick the pecten down by carefully applying pressure to the pectinal spine. Complete the chamber by using a preheated metal spatula to apply melted wax to the unwaxed edge of the platform. The animal is now almost prepared for doing electrophysiology recordings. Now is an easy time to make an indifferent electrode connection. Insert a silver wire between 2 tail segments into the scorpion's hemolymph. Press some of the wire that exits the animal into the clay to secure it and leave enough length to reach the amplifier. Now set the preparation aside for at least an hour before making recordings to allow the preparation to settle. For recordings, use a micropipette puller to make pipettes with an inner diameter of about 2 microns larger than the sensilla pore diameter, which varies between animals. 1 hour before recording, fill the pecten chamber with 5 microliters of mineral oil. Then position the animal beneath a high-powered microscope with long working distance objectives and epi-illumination. To chemically stimulate PEG sensilla, load the stimulant pipette with aqueous tastants mixed in an electrolytic solution using a micropipette filler. Then attach the loaded stimulant pipette to a micromanipulator. Now insert the recording electrode into the open end of the stimulant pipette. It is important to ensure contact between the metal electrode and electrolytic stimulant solution. Connect leads of the recording and indifferent electrodes to a differential amplifier. The amplifier is connected to an oscilloscope to visualize electrical activity and to a speaker to audibly detect electrical change. The setup is now ready for taking recordings. Using the micromanipulator, carefully lower the pipette onto a sensilla pore. A change to higher frequencies will be heard when electrical contact is made between the pipette electrolyte and the fluid within the PEG sensillum. The distinct popping of neural action potentials can be heard at this time as well. The duration of chemical stimulation per pectinal sensillum can vary from as much as 30 minutes to as little as 1 second. Generally, one pipette can sample more than 30 PEGs. Replace clogged pipettes with clean pipettes. Pipettes are easy to prepare, and clogging is not uncommon, so always keep extra pipettes at the ready. It is possible to record from a single animal for several hours in a single recording session before having to change the mineral oil. A single animal can be kept in place in the recording rig for multiple recording sessions over several successive days. For subsequent sessions, reuse the pectinal chamber but replace the mineral oil. Just remove all the old oil with a pipette and add a fresh drop. This helps minimize the presence of residual stimulant, which may have leaked out of the stimulant pipette during the recording session. Here are representative tip recordings to different stimulants. This trace shows a relatively consistent response to 0.01 molar potassium chloride. This PEG sensillum's response to 0.1 molar citric acid begins much more intensely, then tapers down in intensity within a fraction of a second, followed by some diminishing in frequency. In a typical response to 30% ethanol, the trace shows bursts with multiple spikes between short gaps of inactivity. When comparing tip recordings of different sensilla responses to the same stimulant, like 0.1 molar citric acid, the responses are very similar.
While attempting this procedure, it's important to remember to fill the pipette with a water-based stimulant, which doesn't mix with the oil medium and allows you to chemically stimulate one sensillum at a time.
After watching this video, you should have a good understanding of how to controllably stimulate individual sensilla on a scorpion pecten using mineral oil as a medium.